Welcome to this BiofilmQ tutorial. My name is Hannah and today we will have a look at how to analyze microcolonies. I've already loaded the directory here. In previous tutorials you have learned how to do so. And you have also gotten an overview over the different processing steps, as well as maybe already some detailed overview over the segmentation and the parameter calculation. Today we will discuss how to analyze these microcolonies that you can see in an image here. I have two images in my directory. Both of them show these microcolonies. And what we want to know is, for example, how large are these microcolonies? How many do I have in my field of view? What is the substratum coverage? And if these are the kinds of questions you want to answer, you would like to treat each of these microcolonies as an object. That's the first way of looking at these images. Another way would be to think that each of these microcolonies is also a biofilm. And then you could go ahead and analyze each of the microcolonies in detail and calculate parameters in spatial resolution. If this is what you want to do, you can start with the colony separation, which is explained in an additional tutorial. I will just very quickly show it to you here so you can see how you would do that. You will click on Separate Colonies in the Image Preparation tab under Colony Separation. And then this view will appear in which you can adjust the threshold. You can see that the lower I go, the more parts of the images are recognized as biofilm. And you can also adapt the minimal colony size. For example, if I choose 5000 pixel in this case, you can see that only a few of the biofilms are left. You could also, of course, change this value. And if you want to remove individual colonies from your field of view, you can remove them with this Remove Colony button, where you just draw a polygon around your biofilm and it's gone. So once you've done that, you can click on Keep Directory, because in this case you don't have a time series, we can do that, and on Clone Images. And then what happens is that a couple of new images have appeared in the file list here. Each of them is labeled colony 1 to colony 9. And you can see what changes between these images is the cropping. So now, if we were to perform, for example, a segmentation on all these images, it would be performed for each of the different biofilms which were identified in the Separate Colonies view. And then you could do a detailed analysis, you could do a cubing for the biofilm to get spatial resolution, and so on. How to do this is described in the other tutorials that talk about segmentation in detail, parameter calculation, and so on. But today we'll now come back to this first way of looking at the image, where we said that we could also treat each colony as an object in the field of view, and then just calculate a number of statistical properties for these objects, for example, their size distribution, like standard deviation or mean and max value. This is, for example, useful if you do a screening and you want to find optimal conditions for your biofilm growth, then you would do that. And the way to do this is that you first segment your image. There are additional tutorials talking about segmentation in detail, so we will now just very quickly go through them. We will not choose a cropping, nor do a pre-processing. The denoising, we will choose only the deconvolution filter up here with a standard input. So we will uncheck the top hat filter to save some time. For the thresholding, we use Otsu and in two classes, which usually works well. And what's important here is that for the object declumping, we do not use cubes, but instead we set none in this drop-down menu here. Because in that case, the biofilms will not be cubed, but they will still be recognized as separate objects because they can be distinguished through the segmentation. In the post-processing, I can now choose to remove very small biofilms. Maybe it's just a few cells which are attached to the substrate. And here, for example, I can choose 10,000 pixels, which would correspond to 100 cube micrometer about 100 cells, so I can say anything lower than 100 cells is not a microcolony yet. So now I can select the images, the two ones down here, that I want to analyze. I click on selected images to adapt the image range, and then on segment cells to perform the segmentation. Once I have done that, we can move on to the parameter calculation. In this case, you want to calculate structural parameters, for example, the substrate area that each microcolony is attached to. We can also calculate other properties, for example, the local density in each of those microcolonies or fluorescence properties. So this is totally up to you. I think for us, we will do the substrate area and then maybe a local density. And again, we will click on calculate objects to calculate these properties. Once we have done so, we will move to the visualization tab to have a first look at our results. The visualization tab is also explained in additional tutorials. And for now, we will just very quickly load our biofilm and have a look at it. We can see in this file list here, our two files have appeared, the ones that we segmented. We can see the number of cells is 22 in our first image and 35 in our second image. Now number of cells corresponds to the number of microcolonies because each microcolony is recognized as a cell. And then if, for example, we want to plot the distribution of volumes of in one particular image, we can choose the plot type 1D histogram, select only one image, and then choose shape volume as the x-axis. And if we do that and choose minimal and maximal for the x limits, we can see here that we have a couple of microcolonies which are between 0 and 2000 cube micrometers, and then there's one really big microcolony. If we look back at our image here, we can assume that the big microcolony is this one here, and the others, which were typically similar sizes, are the other microcolonies that we have seen in this image. We can now look at any parameter distribution with such a histogram. We can also look at properties over the whole image or the whole field of view. In order to do that, we go to global biofilm parameters here, where we have to choose the 2D XY plot per frame method. And then we can choose frame as the x-axis, where frame is the index of the image. We click on all images to again take both images into account, and now we can choose on our y-axis the different properties that we want to measure, for example, the cell number. And here you can see that in our first image we had a little above 20 objects, 22, so 22 microcolonies. In the second image we had 35. This you could also read from the table here. We can look at the complete substrate coverage, which is like summed over all cells. See that it's a bit higher in the second image. But we can also look at statistical properties of the local parameters, which means of each of the microcolonies. For example, the standard deviation between the volumes of microcolonies. You can see they're approximately equal between the frames, or we can look at the maximal volume. So you can see that with this method, if we had more than 2 images, for example, corresponding to a number of different conditions that we want to screen, with these plots we can see how the overall shape of microcolonies changes. And this can be very helpful when determining, for example, optimal conditions. So this is how you can use and analyze microcolonies with BiofilmQ. And if you want to learn more about how to analyze the individual colonies with spatial resolution and therefore the cubing method, you can have a look at our further tutorials that discuss the segmentation, parameter calculation, and all other processing steps when using the cube method.